So welcome back everybody to episode 14 of the Computomics podcast. I'm very excited today to talk to Emeritus Professor of Plant Breeding at the University of Hohenheim to take our discussion a little bit more into the future of plant science and plant breeding and how the efficiency improvements in the field can really take our food systems and our way of thinking about agriculture into a new direction. I'm really looking forward to our discussion today, so I hope you enjoyed the episode. So, Albrecht, welcome to the episode. I'm very happy to have you here, and thank you for joining us.
Yeah, thanks also from my side. It's a pleasure to participate in this podcast.
Yeah, so I'm really excited to have somebody talking back about plant sciences. We've sort of taken a loop around during our latest episodes. We've talked about metagenomics and we've had a chance to talk about some broader topics. And it's really nice to come back to the core of what we do at Computomics, which is really plant performance and phenotype predictions. And I think the concept of sort of taking a walk around things also is reflected in the way that you came to plant science. If I understood correctly, you started in statistics and then looped back around to agriculture. So maybe you can tell us a little bit about how your career came to be.
Yeah, well, I grew up on a family farm of small size, typical for Southwest Germany. And I was the first to attend high school in my family. In Germany, it's the Gymnasium. And also I was very good in mathematics in school. I didn't dare to study mathematics. So from my background, I started in agricultural biology. And this study had been introduced newly the year before I started.
Interesting. And where was this?
This was agricultural biology at the University of Hohenheim, actually. Yeah, and during my undergraduate studies, it turned out that I really enjoyed mathematics a lot. I also realized, particularly in many lectures, for example in biochemistry or population genetics, that a good command of mathematics would be extremely helpful, especially if I wanted to pursue a career in science. Yeah, during my studies in mathematics, I still kept contacts with my professors in population genetics. And actually, I wrote my master thesis applying probability theory to a problem in plant breeding. In other words, I had always in mind to employ my mathematical skills to problems in biology and especially in plant breeding.
I see. Your detour was always with the intention of coming back. I guess it wasn't a detour. It was a strengthening of a tool that you wanted to use when you came back to the agricultural sciences.
Exactly. That was my intention. I was never interested really in pure mathematics because I felt, using mathematical skills for the numerous problems in biology, that's something which is of interest.
I guess the same way that we feel like using big data and computational power for plant sciences is also, it is about, of course, machine learning and computers and things like this, but it's more so about the end goal, right? And the end goal is improving our agricultural system. So it seems like we had the same idea. Okay, great. So maybe you can tell us a little bit. So currently or always you've worked on maize. Has that been something that you always started working on maize, or is that something that's been sort of your passion in the different part of your career?
Yeah, actually my first contacts with maize were on our family farm, where maize was a rather new crop. It was only introduced in Germany in the 1960s.
Really? I didn't know that.
Because before there were... during the first 15 years of my life, I think I've never seen maize. So, and then that continued when I worked at the university. Then my major professor, Professor Schnell, he had introduced hybrid breeding in maize in Germany. So the first hybrid that was released by my major advisor in Germany.
Wow, that's very exciting.
And a major impetus for my interest in maize came with a stay in the United States in the middle of the Corn Belt, of the US Corn Belt. I had the privilege to receive a scholarship, and I could spend almost 2 years in the mecca of maize breeding, I would say, at Iowa State University in Ames. Now, this was, I think, which gave me the final decision to do my further research in maize, but there are many other reasons which I can explain, of course, to you. What makes maize, I think, very fascinating for a scientist and particularly also for a plant breeder.
Yeah, I would love to hear about that. Tell us, our audience and me specifically, something that's fascinating about maize to you, or what keeps your interest, why you wake up every morning and say, back at it today.
The first reason, I think, for me is, or why I worked with maize during my whole career as a scientist, is maize is one of the most important model plants in genetics and plant breeding. Maize served as a model plant, for example, in cytogenetics, because the chromosomes are large enough so that they can be observed under a normal light microscope. And one of the most significant discoveries of modern genetics, namely the discovery of jumping genes, which are also known as transposons, were made in maize by Barbara McClintock, who earned a Nobel Prize for her discovery.
Wow, that's so great. That touches on so many things that I've worked on. I, of course, started in cytogenetics, so I have a very close passion for it. I didn't know that. I guess, never imagined what a corn genome looked like. That would be interesting to see. And then I always thought about transposons in terms of my Drosophila studies, and then, here you are talking about it in corn. So that's interesting. Keep going. What else?
Yeah, and then maize was, after rice, also one of the first important crop species where the whole genome sequence became available. That happened in 2009. Another reason for me was maize is really a model plant for plant breeding. And the reason for this is hybrid breeding was invented with maize about 100 years ago, and still serves as model crop for this breeding scheme, which is the only breeding scheme that does not occur in nature, but was, so to speak, invented by man.
Interesting. And of course, it's attempted to be transferred to many other crops, right? So the success of it in corn, I guess, in maize.
Now, one of the big advantages of maize, and this is, I think, also the reason why hybrid breeding was invented with maize, is that the two sexes occur on the same plant, but they are spatially separated. You have the tassel on the top of the plant providing the pollen, and you have the silks with the egg cells in the middle of the plant where later the ear develops. And this makes it very easy to perform self-pollination and cross-pollination in a very easy and very effective way. Another reason why working with maize is rather easy is a high multiplication coefficient. On an ear of maize, you have about 200 to 300 seeds. In most other crops, you have maybe 20, 30 seeds. And you can imagine, if you want to evaluate later on genotypes, this is very important that you have enough seeds. That means you can replicate trials.
And each of the seeds is identical on each ear of corn?
That depends on on the genetic material. In inbred lines, the seeds are all genetically identical, also in a single cross hybrid. That's due to Mendel's first law. The uniformity law says if you cross two homozygous inbred lines, what you obtain, the F1 cross, is genetically uniform. But if you would work now with a ear of corn that originates from a plant, from a landrace, and landraces were planted before hybrids came up, then the seeds would be, in our terminology, just half zips. They would have the same mother, namely the plant where the ear was grown, but this plant would be pollinated by the pollen cloud, and the pollen cloud is composed of the pollen from many, many different plants. Yeah, because maize is a cross-pollinated species. The plants release the pollen to the air, and the wind, so to speak, mixes the pollen. And for that reason, in nature, pollination is cross-pollinated, and for that reason you get a mixture, so to speak, of different pollen.
Of course, yeah. It makes sense. It kind of makes me sad that we're missing all of that diversity, and a little bit nostalgic for some reason. As I imagine the pollen flying through open fields, I don't know. There's a nostalgia that's no longer the way that we grow plants.
But there is another point to this now. And that's another reason for me to work with maize. Namely, the tremendous genetic diversity that we have available in maize. Just what you were mentioning, yeah. Maize breeding is concentrating on two races of maize, and altogether we have around 250 races in maize. So we use only about 1% of the total genetic variation that is available in maize.
Amazing.
And that's, in other words, we have still a tremendous reservoir available that we can use for breeding.
And is that where you imagine the field moving, or where do you imagine to be the future then?
Yeah, I think that's one topic of research currently, namely to try to exploit the genetic variation in landraces. Landraces were the populations of maize that have been cultivated before hybrids were introduced. And for example, in Europe, we have currently in the seed banks about 4,500 landraces stored.
Wow.
If you go to the US, you have around 30,000 landraces stored in the seed banks.
And where, I'm just curious, who's responsible? Where are the seed banks in Europe and the US? Do you know?
In Europe, they are in different countries. In the US, the biggest seed bank is in Fort Collins. And another one, a very big one as well, is in Mexico City, which is maintained by the CJR Institute, CIMMYT, which stands for Center for the Improvement of Maize and Wheat. And they store about, again, 30,000 landraces.
Okay. Sorry, I took you off topic. Continue.
Yeah. So, I mean, that's of course very important also for breeding, that you have enough genetic variation available. Now, a third reason for me to work with maize was that hybrid seed can be marketed by companies, and it's sold every year anew, which is, for example, not the case in Germany with line varieties. With hybrids, farmers buy the seeds every year anew.
Right.
And that gives, of course, the companies the opportunity to earn money, and for that reason they invest also in research a lot. Another point is why companies are very much interested in modern research is that in hybrid breeding you see the varieties only at the very end. So you need a lot of theoretical tools to optimize the breeding scheme. And that was, of course, for me as a mathematician also very attractive.
To apply those probability
these-
statistics and
Yes.
of course.
Yeah.
Is that why you're so interested sort of, or feel so engaged or optimistic about, sort of our approach to predictions, because you understand the mathematics behind it, or you're deeper into the mathematics of it, and that sort of gives you a richer understanding of
Yeah.
how everything comes together?
Yeah, that's, I think, exactly one of the topics I'm very much interested. And of course, I'm interested about the tools that computomics can offer to improve our abilities. And maybe I can explain you where these tools come in.
Oh, that would be fantastic. I would love that.
And maybe I give you a very short excursion of maize breeding. Maize
Yes.
or hybrid breeding has two essential components, very simple ones. The first step is you have to develop pure inbred lines. And that was traditionally done by recurrent selfing. That means you self the plants or the plant genotype for 6 generations until you get completely homozygous plants. With each step of selfing, you reduce the number of heterozygous loci by about 50%. So after 5 or 6 generations, you have pure breeding lines, homozygous lines. Now, this was a traditional method, and since about 20 years, we have a new method available, which is called the double doublet technology,
Okay.
where you produce the inbred lines in one step. This method is very efficient, and for that reason, the number of lines the companies develop now per breeding cycle has increased dramatically. When they produced, for example, 1,000 lines in the past, maybe 15 years ago, nowadays they produce 10,000 new genotypes every breeding cycle, every year anew. Now again, this is the one component. The second component in hybrid breeding is you have to find hybrid combinations.
Right.
So imagine, and maize breeders work normally with 2 populations, we call them gene pools, with 2 gene pools in Europe. This would be Flint and Dent. In the United States, you have 2 different Dent pools. So imagine you have now in each pool 1,000 new lines available every generation, every year. From these 1,000 lines in pool A and pool B, it is possible to produce, in principle, it's possible to produce 1 million new hybrids every year. And
Seems
you can
easy
imagine
to grow 1 million new hybrids, no?
this is impossible to produce, and it's possible to test these. But we have the task to identify the most promising hybrids amongst these million of hybrids that we can possibly produce. The first point is if it would be possible to make predictions based on the properties of the inbred lines, on the hybrids we obtain from them, that would be very efficient. For example, in our case in the example that we just discussed, we would have to investigate 2,000 inbred lines, and from this information we could predict the 1 million of possible hybrids.
Right.
So
Simulate,
you
let's
can
say.
imagine that is in principle a very effective method.
It's effective if you're able to make any conclusions from it, right? It's not effective in and of itself.
If you can make any conclusions. Now, one problem in hybrid breeding is that the properties of the parents is not a good predictor of the performance of the hybrids. You may have 2 inbred lines that have a high yield. This does not necessarily imply that the hybrid is very good.
And that's in fact the opposite, right? So the surprise of crossing two, let's say, completely average looking lines to produce something fantastic is the sort of, I guess, the high that you get, right? So
Yes.
that's the payout.
And what we are now doing with new technologies, with genomic tools, we genotype these parent lines usually with SNP markers covering the entire genome. We have meanwhile arrays available that have 600,000 SNPs at reasonable costs. So we analyze or we genotype all 2,000 input lines in our example, and we would produce a certain number of hybrids that we would use as a training set. For example, we would produce, let's say, 1,000 of hybrids out of the 1 million possible hybrids.
Right.
And then we would use, for example, methods of machine learning to associate the information from our hybrids that we have tested with the genomic information on our parent lines. And we obtain a model equation, for example. What we are doing is we assign, or we try to estimate the effects of each SNP separately and get a prediction equation. And in other words, we sum up the effects over the SNPs. If we would have 600,000 SNPs, we would try to estimate the effect of each SNP and sum up over the 600,000 SNPs. In other words, that's a purely additive model. Now, if you look at nature does not work necessarily in an additive manner.
That's true.
We know that we have networks. Genes work not individually, but they work in networks. And this is an aspect that has not been considered properly in our current models, where I expect that machine learning, for example, can really add new information that gives us a higher power, better prediction accuracy when we do prediction. And you can imagine that's very important if we phenotype, so to speak, only a small portion of the hybrids that are possible, namely only one per mil. If we would have 1 million possible hybrids, we phenotype only 1,000, then we phenotype only 1 per mil. So we would be interested to make predictions about the remaining ones, namely the 999 per mil. And that would be a very efficient system. And there comes in, for example, the tools that computomics develops.
Right. We get excited about it, and I think sometimes other people get excited about it too. Maybe they get concerned because they don't have that sort of concrete understanding within themselves of how it's really possible, right? So we may say all these things, but to somebody who's never done these sort of predictions themselves, it's hard to really conceptualize the validity of it or the potential in it. So how would you explain this to somebody who's maybe hesitant and says, well, we've tried associating genomes to phenotypes and it hasn't worked out. For example, we found a gene and then we put it in the field and then it all sort of went sideways and we just don't believe that this is possible. Do you feel like you meet a lot of people like that, or?
Yeah, the question I often obtain is about, particularly in Europe, is of course genetic engineering. Now, I should emphasize what I was talking about. These are complex traits.
Of course.
In other words, traits where thousands of genetic loci are contributing. And for that reason, it's also more a statistical approach. If you work just with a single gene, you can follow its effect relatively easily. Yeah, but I'm coming more from the quantitative side, from quantitative genetics, and for that reason, so to speak, I'm interested in the sum of the effects of all these 1,000 genes or more that influence characters like yield.
Sure.
I'm asked about genetic engineering. The other topic, prediction, is not yet discussed in public. Maybe it will be discussed when personalized medicine, when tools for personalized medicine are developed, because essentially the same principles lie behind personalized medicine. Actually, what I should mention is the prediction equations that we use in plant breeding, and the ideas have not been developed in plant breeding, unfortunately, or regrettably, I have to admit. They were developed by animal breeders. But since genetics works essentially the same, you can apply the same principles to breeding of animals, breeding of plants, even microbes, and you can use it for predicting complex traits also in humans. In humans, it's a little bit more complicated because you have confounding effects, environment, etc. In plant breeding, we are in the lucky situation that we can perform designed experiments.
It's interesting. I guess it's amazing how the same storylines get carried through, from us to then livestock, to then crops, and like we are sort of all tied together in a very beautiful, connected way, right? If we choose to think about it, different ratios, but sort of the same storyline. So yeah, that's wonderful. Well, thank you so much. I really appreciate your time. Thank you for giving us a great primer on maize genetics and on ways that we can integrate our technology into this. And I really look forward to speaking again. Thank you everyone for listening to this episode on maize breeding with Professor Melchinger. I hope you enjoyed the primer on breeding and his discussion on how machine learning can improve our accuracy when predicting phenotypes. I know it was a little bit longer episode than usual, but I think it was deeply informative and I hope you enjoyed it. Please leave a rating or review and get in touch with us, info@computomics.com, and we'll see you next time.